Hello, class. Welcome to Human Physiology, Biology 12 at Saddleback College. In this very first lecture, we will talk about some key terms that will follow us throughout the semester, and I will also demonstrate how some of these key terms can be applied to a specific example in our body called thermoregulation. There isn't a lot to cover in this lecture. Like I said, I'll start out with some key terms, and then I'll give you an example of homeostasis in our body, specifically with regards to thermoregulation. You are here to learn about physiology, which is the science that is concerned with how the normal living organism and its components work. It's very boring, if you ask me. It's very boring to know about skeletal muscle contraction. How our body is supposed to work is boring. Most of you are not in this class to learn how it works. Rather, you are called into this pathway because many of you want to be a healthcare provider of some sort. Most of the people you're going to be treating in the future are not going to be healthy individuals. Very rarely does someone walk into a hospital and say, hey, I just want to stay in bed for a week and have you bring me my meals. Arguably, the closest person to that who would check into a hospital who is relatively healthy would be a woman who is at the end of her pregnancy. Most of you are following this career path because you're interested in the disease state, which is pathophysiology. You will be getting a lot of both in this class. You will learn the normal physiology, and then you will learn about the disease state. The disease state is the cool part, and arguably the reason why most of you are in this class. So one of our first terms that I want you to know about is homeostasis. This was first coined by a French physician, Claude Bernard, and he referred to homeostasis as a milieu intérieur, which means a stable internal environment. And when he started to characterize this phrase, he really identified that we were talking about the plasma, specifically about the proteins in the plasma. And then by extension, the proteins that are found in our 100 trillion cells. It is the proteins in our plasma and in our cells that help our body maintain its optimal conditions for function and survival. They are the workhorses of the cells. So we want to prevent the proteins from being denatured. That means unraveling from their folded active state into an inactive state. Some of the terms that go along with this phrase, milieu intérieur, would be 1, a variable. A variable is anything that can be measured in the body. For example, we can take our patient's temperature. We can draw blood and find out their blood pH, the sodium concentration in their plasma, their glucose concentration in their plasma, their blood pressure, their urinary output. Anything that can be measured is a variable. These variables are maintained at a set point. And those two words make a student think that we are held and locked at a particular level. For example, blood pH. Optimally, it is 7.40. That would be identified as a student as a set point, when in actuality, it could be called set range, an optimal range. So our optimal range for blood pH is really 7.35 to 7.45. So instead of set point, think an acceptable range. Deviation is a change from the set point when you are going beyond the normal acceptable values. Correction, also known as compensation. How much correction do you need? Depends on how much the deviation was. The greater the deviation, the more the correction necessary. Speaking of types of correction, there are two types that I'd like to discuss: negative feedback and positive feedback. Negative feedback, simply put, means that the deviation and the correction go in opposite direction as shown down here at the bottom of the slide. There needs to be some sort of way to detect if the variable has in fact deviated from the set point. You need a sensor. Oftentimes this is going to be some sort of receptor, some sort of modified nerve, if you will. Then the input signal has got to be sent to the brain or spinal cord. These will be integration centers. And those integration signals or centers are going to refer to the set point and the incoming signal and figure out how much of a deviation, if at all, has occurred. Then we're going to have a correction that's triggered. This will be a pre-programmed response. That is according to the genetic ability of that particular cell. For example, the heart will respond like a heart will in response to, let's say, the need to increase blood pressure. The heart would beat with greater vigor, with greater strength. But blood vessels will also help increase blood pressure. They would increase blood pressure by increasing the vascular tone, by increasing the amount of smooth muscle contraction. Excuse me. So that's what I mean by the pre-programmed correction, the gene expression possible by those cells. Then the output signal is what's sent out by the integration center to the effectors, the parts of the body that can actually do the correction. In my previous example, that would be the heart and the blood vessels with respect to increasing blood pressure. And the idea is that now the deviated variable can return toward set point. Positive feedback promotes change in one direction. In other words, deviation and correction go in the same direction. And this might be perceived as a misnomer to students, thinking, well, how are we correcting anything? If deviation and correction go in the same direction, aren't we getting more deviation? And the idea is when we use positive feedback in our body, it is to promote a drastic change, an avalanche-like type change in the body, as shown in my top right picture. You want a snowball effect. Something needs to be modified in the body very drastically, very quickly. For example, most of the time we want our blood to be maintained in a fluid state, a liquid state, which is easy for it to be pushed around the body by the heart. But there are times when we have damage to blood vessels, we have cuts, for example, and we appreciate if our blood can drastically convert from a liquid state to this gelatinous state that we call a clot. Now, when that happens, we have platelets that become activated that lead to a clot formation. And that clot in turn leads to more platelets being activated, which leads to more clot. So you should be hearing activated platelets leads to clot, which leads to more activated platelets, which leads to more clot, more leading to more. Deviation and correction going in the same direction. So we get a very rapid change. Now, ultimately, all positive feedback corrective mechanisms need to be stopped. Ultimately, we got to get back toward that set point. So all positive feedback mechanisms must be ended ultimately by a negative feedback mechanism. If this can't be done in a healthy way, then the ultimate negative feedback to stop the positive feedback would be death. And for this reason, when we see positive feedback happening in the body, we frequently see an example of this when the person is in a diseased state, in particular with heart failure. And we will talk about heart failure being a positive feedback diseased state when we get to Unit 3. Another example of positive feedback would be labor, shown here, well, the woman in the bottom right picture being terminally pregnant with the parasite that she is carrying. When the fetus is large enough, it starts to push on the cervix. This leads to neuronal signals sent up to the woman's brain, specifically the hypothalamus, to release oxytocin. This leads to more uterine contraction, which leads to more of the fetus being pushed, and if its head is down, the head will push on the woman's cervix. This leads to more oxytocin, which leads to more uterine contraction, which leads to more cervix stimulation, which leads to more oxytocin, and you're hearing more leading to more, until ultimately the parasite is birthed, and that would be your ultimate negative feedback, meaning now the baby is born, there is no more stimulus on the cervix, and the release of oxytocin can start to decrease. All cells participate in this regulatory effort in the body. Both positive and negative feedback mechanisms can occur down to the cellular level. This can be applied to regulation of gene expression, which genes are turned on, and in what quantity and when. It can also be applied to tissues, which is a group of cells with a common function, to organs, which is two or more cell types working together, even between organ systems. An example of how organ systems can work together to maintain homeostasis is shown here on this slide. Every organ system has a job to help the body maintain homeostasis. If you think about your renal system, the kidneys are a huge component of that. They filter your plasma multiple times a day to remove waste products. A buildup of waste products can easily cause the demise of cells. The respiratory system, we think of the lungs and the skeletal muscle diaphragm causing ventilation of the lungs, and we think of gas exchange. The cardiovascular system with the heart as a pump to physically pump the blood around to deliver new nutrients and pick up waste products from the cells of the body. The GI tract and accessory organs like the liver to help us with chemical digestion, even mechanical digestion of the food we eat, the absorption of the nutrients, and anything that we don't absorb must be expelled from the body. The musculoskeletal system even helps maintain homeostasis. We need to get up and walk towards a food source. We also need to get up and walk towards the bathroom to relieve ourselves of our waste products. The nervous system and endocrine system also are important for maintaining homeostasis, and those two can oversee all the other organ systems and help maintain control of them. So we're going to see a common theme throughout the semester. We're going to see that there is local control within an organ. Then there is also going to be an overlapping control from the nervous system and endocrine system on a particular organ system. Let's evaluate how the nervous system and endocrine system can help maintain homeostasis. How are they similar and how are they different? So first thing, both the nervous system and endocrine system are common in that they both use a chemical signal. The nervous system uses a neurotransmitter and the endocrine system uses a hormone. The nervous system releases the neurotransmitter directly on target cells through a functional contact called a synapse. The endocrine system releases the chemical signal into the bloodstream, and this is distributed throughout the body. The time frame over which these two systems work is very different. The nervous system is quick to fire and release a neurotransmitter, but this neurotransmitter has very short lasting effects. Usually we're only talking about 1 to 10 milliseconds. The endocrine system is slower to respond. It can take seconds to minutes to days to be provoked, and it can last for a long time. Some hormones can have lasting effects for over a month, for example. There is a price for maintaining homeostasis. The price is cellular energy, ATP. When we think of homeostasis, I want you to understand that that is really a steady state, a stable internal environment. This is not equilibrium. For example, most of your potassium is found within your cells. Most of your sodium is found outside of your cells. Clearly, this means there is a chemical gradient. Potassium wants to diffuse out of your cells into the extracellular fluid, and sodium would prefer to diffuse from the extracellular fluid into the cells. If they were allowed to diffuse, then they would reach equilibrium. There would be equal amounts on either side of the cell membrane. This is not how our body functions. In fact, if these two electrolytes did reach equilibrium, I would say you'd be dead. We cannot have them reach equilibrium. We need those chemical gradients, high potassium inside the cell, high sodium outside the cell. That is our steady state. When we have studied these types of corrective mechanisms, we have found that many corrective mechanisms work together or synergistically. We have also found corrective mechanisms that are antagonistic to each other. And these corrective mechanisms can be utilized in our body at the same time. That's easy for a student to appreciate and learn when they are synergistic, working together, but it's harder to tease apart when we have opposing corrective mechanisms on at the same time. It will take a lot of practice for us to get these things right as we go through the different organ systems for this semester. We have also learned a lot about these corrective mechanisms through knockout mice. That's what KO stands for on this slide, knockout mice. We have obliterated some of these corrective mechanisms in mice by deleting genes. And when the mice developed normally and had normal lives, we realized that there must have been other genes, other corrective mechanisms that worked in a synergistic way that provided redundancy. In other words, backup plans. And that's another level of complexity that we'll have to deal with as we move on throughout the semester. It does complicate things because that means there's going to be competition or a hierarchy of these corrective loops. So again, the energy is the price we pay, the cellular energy, ATP. Now I'm moving on to a specific example of homeostasis called thermoregulation. And the question I want to start out with is, are we locked at our set points? And the answer is no. We are constantly deviating. Think about your body temperature right now. Do you feel cool? Do you feel warm? Were you even thinking about it before I made you notice or think about it? Probably not. In the top picture, you're seeing that our body temperature is held at a pretty constant state, steady state, despite us being exposed to very different atmospheric temperatures. That is homeostasis. We are never locked at a body temperature. We are constantly deviating. We are getting a little too warm, and what do? We cool off to adjust. Then we get too cool, and then we warm up. But over time, if I were to average your body temperature over the day and average it out, it would be relatively stable relatively at a steady state with very minor erratic swings. This is because we are homeotherms. We have the ability to regulate our body temperature. Poikilotherms, shown here as the lizard, cannot regulate their body temperature. Poikilotherms must come out of their den when the sun rises. They find a warm, sunny spot, and they absorb the radiant heat from the sun to increase their own body temperature before they become active. I'm sure some of you have been on an early morning hike only to come across a snake in the trail that didn't move as you approached it, didn't make any quick evasive maneuvers because it was too cold to move. That's why it was out in the sun on the trail. Newborn infants can be thought of a lot like a poikilotherm. First of all, they look a lot like a lizard if you ask me. But I mean that kind of tongue-in-cheek in that they do not have a mature hypothalamus, which is the part of the brain that helps regulate our body temperature. Newborns have immature brains, and their hypothalamus cannot regulate their body temperature quite well, which is why we're always fretting over a newborn to be swaddled and underneath blankets to help maintain the body temperature. Your body temperature also fluctuates throughout the day. You are coolest in the wee hours of the morning, and you're warmest in the late afternoon. Clearly, body temperature can fluctuate based on physical activity. And if you're a menstruating female, you run warmer when you are just shortly after your ovulation. Now I would like to address ways that we can lose heat, and some of these you can also gain heat by. So first of all, the primary way we lose heat is through radiation heat loss. This is from us emitting infrared heat waves. This is you basically trying to warm up every object in the room, everything. A lot of students are confused by what radiation heat loss means. So let me choose an inanimate object. If you've ever been near a fireplace or a wood-burning stove, you have felt the heat coming off the fireplace or the stove. You weren't touching it. You might have put your hands next to it and you could feel the heat coming off. That's the radiative heat loss from the fireplace and the stove. If I get close enough to you, I don't have to touch you, but I can feel the heat radiate off of your body. The next way that we lose heat is through evaporation. Evaporation first begins with sweat. Now, sweating itself does not cool our body. You can get a layer of moisture on your skin, but if you are in an environment where there's a high humidity index, then the water that's in the liquid phase on the surface of your body is not going to be very likely to evaporate, go into this vapor phase, and enter the atmosphere. So if you have ever been sweating profusely, but it didn't evaporate well, let me tell you, you were not cooling off at all. Conduction and convection heat losses go together. Conduction in a certain way, and I'll explain how in just a moment. Conduction is the physical transfer of heat from your body to something else through physical contact. This can be from you touching a counter or the toilet seat or your phone. If you were on your phone for 20 minutes and I picked up your phone to use it, it might feel warm on my ear. I'm sure you have had some circumstances where you went to a public restroom, sat down on the toilet, only to find out that it was warm. And that might have made you stop and think, what took the person so long that was there before you? Many textbooks say conductive heat loss is the physical contact of your body with some object, a solid object, and you transfer heat to it. It does not have to be a solid object. You can be immersed in water. You are in physical contact of water. That is not solid, that's a liquid. And you, right now, you're immersed in air. It's weird to think. You are touching air. Your physical contact with the air around you is going to lead to heat transferring from your body to the air around you. Then what does hot air do? This is where the convection occurs. Hot air, warm air, rises. When it rises, then cool air is replaced, and you're going to repeat this process by heating up the air around you. So again, radiative heat loss is through infrared heat waves emitted from your body. This is different than conduction, our physical contact with the immediate layer of air around us. If there is a wind index, you might have heard about this on weather channels, then that means the convective heat loss from our body is augmented by a breezy day. That's the reason why layers help. They prevent the convective heat loss from happening too precipitously, too quickly. So again, these are ways we lose heat that I've just talked about. But can we gain heat from some of these? Of course, we can have radiative heat gain. We can have heat gain from sitting close to that wood-burning stove. We have radiative heat gain when we go outside and the sun is beating down on us. So we can also have heat gain from radiation. We can also have heat gain from conduction. For example, if you were to put your hand on a hot stove, you would gain heat from that physical touch. So you should be able to identify these mechanisms for heat loss, but also appreciate if we can use the same mechanisms to gain heat. Now we will continue the rest of this lecture in part 2 of this segment.